morning, everybody. At the outset, I welcome you all to the distinguished talk on COVID-19 Lessons for Science and Society. And today we have with us a distinguished resource expert, Dr. Debprasad Chattopadhyay, who is the Director and Scientist G of ICMR, NITM, Belgavi. I welcome you, Dr. Chattopadhyay, sir, for this session. Welcome to you.
Thank you, sir.
We have with us session chair for this talk, a distinguished academician and scientist, Professor Kushal Das, distinguished chair professor, laboratory at Vascular Physiology and Medicine, BLDE University, Vijayapura, and is also the Dean of Environmental Health under UNESCO Chair, Life Sciences. Welcome to you, sir. We have with us Honorable Member of KSTA and former DG of NACA, and he was also the Director of ICR, Dr. Eknath Ambekar, sir. Welcome to you, sir, on behalf of all of us. Senior academicians, including Dr. Satnoor, Principal, scientists, teaching faculty, students have joined for this session. Welcome to you all. Now let me have the privilege of introducing session chair, Professor Kushal Das, sir. A brief overview of his accomplishment. Professor Das did his PhD in physiology from University of Calcutta, and his doctoral research was entitled Metallic Pollution, its effect on male reproductive system, with special reference to protein inadequacy. Professor Das' research on experimental biology for progress on low oxygen microenvironment in rodents, especially in chronic, sustained and intermittent hypoxia in experimental animals treated with heavy metals. He has done extensive research on regulation of vascular, endothelial and smooth muscle integrity. Currently, he is working on isolation of specific bioactive compounds from indigenous plants of Karnataka. Next week on Thursday, he will be delivering a talk on this particular topic. And also, he is synthesizing analogs of bioactive compounds to develop drugs against stroke. He has received a number of prestigious awards for his research, including Government of Karnataka's Dr. Rajaramana State Scientist Award, and he has received many awards. I won't be able to tell in a brief time. With this brief introduction, I welcome you to you, sir, and also to take over the session and continue the proceedings. Once again, welcome to all of you. Thank you.
Thank you.
Please take over.
Thank you very much, Professor Ramesh, and distinguished Professor Eknath sir and other distinguished participants in this meeting. It's a great pleasure to introduce Dr. Debprasad Chattopadhyay. He's a very illustrious scientist in his own domain. Dr. Chattopadhyay is a junior to me in academic, as such in the university degree concerned, I'm senior, and he did his extensive research and his academic studies in the Jadavpur University in the biotechnology. His outstanding work on the traditional medicine is remarkable in the aspects of developments of various fields. In fact, he is one of the pioneers, although professionally he is a virologist, and he's a renowned virologist by himself and molecular biologist. He has an extensive experience throughout the world, including Japan. So it is very difficult to say a few words about such a distinguished scientist in the career of my own. And I'm always feeling proud that my junior is in such a level of knowledge and wisdom. And specifically as a human being, he is an extremely good human being that is and very kind towards the students. And his attitude towards the subordinates are extremely beneficial to gain the knowledge by scientists of others. And that's the reason he was very popular when he was the senior scientist at National Institute of Cholera and Enteric Disease, where he most of the time he spent as a scientist of NICED ICMR in Calcutta. Later he transferred as a director and took over as a National Institute of Traditional Medicine as a director. He has a vast experience in the publication in the wide range of journal, including Journal of Immunology, even Nature Medicine. So I should not speak too much about this one. Let us listen a very important topic by which he has actually done extensive work, specifically analyzing the genome sequencing of the COVID-19 virus last year. So he will be speaking in general, the topic COVID-19, a lesson for science and society in his own way. He's a great speaker as such. So let us listen what Dr. Chattopadhyay speak about the science, including his domain as a virologist. Over to Dr. Chattopadhyay, sir. We are honored you are here with us.
Very good morning to all of you. First of all, I convey my sincerest gratitude to Professor Kushal Das for such a kind word. My thanks to the chairman, Professor S. Ayappan of Karnataka Science and Technology Academy, Dr. A.M. Ramesh, CEO of the Academy, Professor Das, Dr. Eknath sir, and ladies and gentlemen. Today's topic you already know. Now this topic is divided into two broad areas. One is the relation, what we learned from this pandemic. That means what science learned from this and what society learned. So broadly, it has been classified or categorized into two sectors. First of all, I will describe about the COVID, because everybody knows about what is COVID. It's a coronavirus disease identified in 2019 first. The virus belongs to a family called as Coronaviridae of the order Nidoviralis, and it has a diameter of 65 to 125 nanometer. The important thing is that this is an enveloped positive sense single-stranded RNA virus. It means that this single-stranded positive sense RNA acted messenger RNA itself within the host cell. Now, coronaviruses are classified into four categories, four subgroups rather: alpha, beta, gamma, and delta. Gamma and delta belongs to animal and bird viruses, but alpha and beta has got to altogether 7 viruses which can infect human being, and all these viruses are of zoonotic in origin. Now, this infectious disease caused by the virus termed as severe acute respiratory syndrome, coronavirus 2. The disease was first identified, as you know, in the capital city of Hubei province of China, Wuhan, in December 2019, and spread globally, resulting in the ongoing 2020-2021 coronavirus pandemic. Till date, it infected more than 16.55 crore people around the globe, with 34.31 lakhs deaths across the globe, spreading over 217 countries until today. Till date, 10 vaccines, 3 in India now, are in emergency use, but there is no specific antiviral drug. Now, COVID-19, biology of the virus and the host. Actually, you know about 3 terms. One is outbreak. Number of disease cases more than anticipated. If increase in an endemic area and not controlled, can be of epidemic nature, is called as outbreak. Then if the disease that affects a large number of people within a region of community and population, called as epidemic, and when the epidemic is covering the entire globe or a very wide area crossing the international boundaries, affect a large number of people, called as pandemic, pan-endemic. Now, what lesson we are going to learn from the part of science? What science has learned from this virus and host interaction? Remember, this is the first virus which caused pandemic, first virus of corona family that got create havoc all over the world. Now, you know, viruses are the very vital member of the web of life, which challenge the very concept of living. It's very difficult to define or to produce a definition, to generate a definition. In 1997, we have put forward a definition of viruses. They are ultra microscopic, are cellular, metabolically inert nucleoprotein particles with or without a lipid envelope and either contain or DNA or RNA or DNA as their genetic material. By characteristic, they are obligate intracellular parasites, utilized host cell machinery to propagate, and they cause ailments as benign as a wart, as irritating as a cold, to as deadly as the bloody African fever. Some can spread easily like coronavirus and kill swiftly. Mostly they have no cure, or many of them have no vaccines also. Now they have numerous invasion strategies, and their host specific, root specific, target tissue targeted or tissue tropism. They have tissue tropism. Each strain has its own unique configuration of surface molecule that enable the virus to enter the host cell by precisely fitting their surface molecule with the molecule of the target cell. And in evolution, they have several advantages. They have genetic variation, DNA and RNA, single-stranded DNA, double-stranded DNA, single-stranded RNA, double-stranded RNA, plus sense RNA, minus sense RNA. Their variety of transmission, various route, and efficient replication within the host, and some have the ability to persist within the host, like HSV or herpes simplex virus, like HIV, and in some cases other viruses also. As a consequence, the virus is adapted to all forms of life and occupied numerous ecological niches, and they cause the widespread diseases from livestock to human to plants. Now, coronavirus. So they are positive sense single-stranded RNA virus with crown-like appearance called as coronum or crown due to presence of spike glycoprotein on their envelope. Their genome characterization revealed probably they are derived from bats or rodents and are the gene source of alpha and beta coronaviruses. However, coronavirus is supposed to be spillover from bat to human via an intermediate animal, pangolin. They are round to elliptical or sometimes pleomorphic in shape with about 60 to 140 nanometer diameter, and they are very sensitive to ultraviolet light and radiation, because always remember the definition of the virus. They are metabolically inert acellular nucleoprotein particles. And coronaviruses use ACE2 or CD147 receptor in human being as a route of entry. I will tell later on these things. Actually, what is ACE2? Angiotensin converting enzyme. Kushal can tell many more about it, but it is a part of renin-angiotensin system that controls our blood pressure by regulating fluid volume in the body, and it covers angiotensin 1 to the active vasotranscription or vasoconstrictor angiotensin 2, thus indirectly increases the blood flow or blood pressure by vasoconstriction. So ACE2 inhibitors are used to treat cardiovascular diseases. That's why it is difficult to design a drug against this ACE2 binding of this virus. I will come later on during the later part of this talk. Now, initially the virus was animalistic because of its animal origin, and as it loses the host, so it was aggressive due to loss of host to find out a new host. Now the situation has changed, and the major events, I am not going to repeat. The origin is from bat, and from bat there are actually 3 viruses are originated. One is through camel, intermediate host, called as Middle East Respiratory Syndrome coronavirus. Occurred in outbreak takes place in 2012, and SARS coronavirus. So one outbreak was 2002-3 from China again. Civet cat is the intermediate host, and third one is the SARS coronavirus 2, probably pangolin is the intermediate host. The work is still going on. Now, what are the characteristics of this virus? The genome of this virus is about 30 kilobase, which encodes 4 major structural proteins: spike protein, membrane protein, envelope protein, and nucleoprotein. Now, the spike protein, it facilitates the entry like a launching pad into the target cell with its three parts. One is a short intercellular part, intercellular tail. Second is the transmembrane anchor. And third is a large ectoderm consisting of two receptor domain, receptor binding domain and membrane fusing domain or subunits. This is very interesting because any other viruses do not have such specific or elaborate structure for launching. Now, sequence analysis of S protein genome showed that coronavirus 1 and 2, they have 75% homology with their spike protein. Receptor binding domain or motif is S of S protein in S protein is conjured between coronavirus 1 and 2, suggesting that coronavirus strain use the same receptor for entry to the host cell. Now, the incubation period is very varied, 2 to 14 days, medium is 5.1 day, and incubation period in sometimes is found that 24 days also. So the life cycle of this virus is still work is going on, and still we need to have a number of data, number of evidence. We are waiting for some more evidences to describe this virus or to control this virus in effective manner. Now, you all know the clinical presentation. I'm not going in detail, and the transmission cycle I already told, bat to human being via the pangolin and then man to man. Now, infection and pathology, what we learned from. There are four parts. You can asymptomatic, pre-symptomatic, symptomatic, and undocumented. Now, combating the SARS coronavirus lies in preventing its spread. But how? When people are unaware about their infection. What are the difference between asymptomatic, pre-symptomatic, and symptomatic cases? A person when is RT-PCR positive for the virus but don't feel unwell, this is called as asymptomatic case, may develop symptom later, but at this stage of asymptomatic time, the person is contiguous before developing symptom. At this stage, the virus can be passed on from one to other. It is unclear on how many transmission happen from asymptomatic cases because it is very varied. Understanding the transmission of virus from asymptomatic carrier is very much required because it requires widespread testing and more data to generate. Many cases, the infected person has a very mild form of disease and common symptom, cough, fever, tiredness sometimes, which may not be noticeable or atypical. So tracking and contacting infectious carrier is a key tool in this case. Now, pre-symptomatic is the infected people are incubating the virus without showing any symptom. Now, the time between catching the virus and showing the symptom can be designated as pre-symptomatic phase. Now, this is much common than the asymptomatic one. About 75% of people who test positive without symptom are turned out to be pre-symptomatic, displaying cough, fatigue, fever, may or may not other signs in follow-up examination. Now, there are other categories. Some cases picked up through tracing are silent spreader who later may develop symptom. Some people can test positive for the virus 1 to 3 days before they develop symptom. So asymptomatic and pre-symptomatic cases are the silent spreader, and that create the havoc for transmitting this virus. Now, you know about the symptomatic cases: fever, cough, shortness of breath, persistent pain, chest pressure, and sometimes in an acute condition also. Now, there are some cases which we do not know about, new things learned by the science. Alongside with silent spreader, there are cases of non-documented infections who have symptoms but don't get tested for any of the reason I do not know. The volume of undocumented cases is much harder to get than the documented one, where people are identified as they having the disease because they sought healthcare or tested through contact tracing. Now, there are several testing protocol. Already I have seen that a few days back you have a lecture on testing of COVID, mainly direct testing and indirect testing. Direct testing gold standard is the RT-PCR, and there are rapid antigen test also. And indirect testing do not very useful for this particular virus. I will tell later on in my next part of the talk. Now, how the infection takes place. The virus enter either via mouth or nose directly to the goblet and ciliated cells, which are abundantly present in the respiratory tract. And these cells are abundantly contain ACE2 receptor. The virus then after entering into the host, it hijacks the host cell machinery to propagate, to produce the progeny viruses. Infected person may be symptomatic or have very less symptom or very no symptom actually, and they may release the large number of viruses in the first week of infection. If our immune system unable to fight back the virus then goes down the windpipe and infect lung. Now, lung contain two types of cells, alveolar cells having pneumocyte 1 and pneumocyte 2. Those are rich in ACE2. Thin type 1 cells are responsible for gaseous exchange, whereas the harder cells of type 2 are secreting a chemical that helps not to adhere the type 1 thin cells each other. So the immune system then stimulates WBC to release inflammatory cytokines that bring more cells to kill in the infected area. Then fluid accumulates and fluid and dead cell accumulates in the alveolar region and leading to the reduced gas exchange. Thus, the infected person needs the oxygen support. Now, to describe in a detail, I'm going to first say how the virus attaches with the ACE2 receptor. These are the ACE2 receptor expression. The virus attaches with the host cell ACE2 receptor and then undergoes endocytosis. First is bind to the ACE2 receptor, and the receptor contains S protein actually of the virus. That S protein of the virus, I already told, has two domains, S1 and S2. Now S1 is the attachment and S2 is now going for fusion. Actually, it undergoes conformational change and that leads to the fusion with the virus envelope with the host cell membrane in the endosomal pathway. Then the virus releases, in the third point, virus releases its single-stranded positive sense RNA partially. That RNA now goes for partial translation, translated into viral replicase polyprotein 1A and 1AB. Now transcription of polyprotein leads to subgenomic RNA synthesis, and RNA translated into S, M, and E proteins taken to host cell endoplasmic reticulum. Now these all things combined with the nucleocapsid N and goes to the Golgi. Now post-Golgi processing, all incorporated into mature virion and traffic to the cell membrane to exocytosis, and ultimately it comes out from the cell as a newly budding coronavirus particle. Now, to establish the infection, the virus has to overcome the host defense barrier and non-specific innate immune system first. Then it releases interferon 1 to attack the natural killer cells for killing infected cells. Now, coronavirus suppresses non-specific immune response and direct the host cell to produce viral peptide protease, membrane protein, which help the virus to mature and release new particle and suppress the development of antiviral immune response of the host called as IFN1 or interferon 1. How these things knows the PL-Pro by inhibitor GL10167 actually cleared these events, and the work is going on to develop antivirals against this virus. Now, PL-Pro of coronavirus also clips off interferon stimulated gene 15 by cellular protease more than other SARS viruses. That's why it leads to a greater inhibition of TNF1 production, TNF alpha production, and that's why the problem is much more. There are several work is going on this line. And now how it happens to be, this is a normal lung, and in normal lung, gaseous exchange takes place with the pneumocyte 1, and this is the structure of alveoli. Now what happened when the infection goes on, SARS coronavirus viral spike protein binds to the ACE2 receptor on alveolar type 2 cell first and inject its RNA. The viral RNA then take control of the infected cell to produce numerous virus copies. The progeny virion, when they released into the alveolus, they infect nearby alveolar cells again. And in severe, moderate to severe cases, what happened with the immune system? Just narrate these things. In immune response, after infection, first virus kill the type 2 cells, which release the chemicals that helps not to adhere, that helps the not to adhere the pneumocyte to one cells. So after the infection, type 2 cells release inflammatory signal to recruit macrophage. Macrophages releases cytokines. You know, normally what happened, I will tell you later on also, when macrophage engulf the virus, it break down the virus into multi-particles, and those particles are exposed in the macrophage boundary well, and T lymphocyte recognizes them and then do the havoc actually. Now macrophage releases cytokine, cause vasodilation, allow more cells to reach into the injury site, and in this stage symptom may start. Now fluid accumulation starts within the alveoli. Now that fluid diluted the surfactant which is released by type 2 cells or type 2 pneumocytes. Now this surfactant dilution triggered the alveolar collapse and reduced gas exchange, because first of all the thin pneumocyte cell one, they overlap each other, so breathing problem comes, as well as the accumulation of the fluid within the alveolar cavity increases. Now the neutrophil recruited at infection site to release reactive oxygen species to destroy the infected cell, but altogether they produce a fibrillar network and called as netosis. And collapses of alveoli due to damage of type 1 and type 2 cells leads to acute respiratory distress syndrome, and in this case hospitalization and intensive care is required. And in severe inflammation, protein-rich fluid enter bloodstream and travel to the whole body, thereby attack other cells also, because most of the cells in our body contain ACE2 receptor abundantly and causing systemic inflammatory response syndrome or SIDS. Now SIDS may lead to specific septic shock and multi-organ failure to death, but intervention in proper steps can prevent the death. Now this is actually what happened. In the left side is a healthy lung, you see, and in the injured lung, what happened? After the entry of the virus, it releases interleukin 6. And you know interleukin 6 can able to damage the liver. And starting from the liver damage, other organ damage takes place, as well as a cascade of event takes place. And cellular debris accumulated, so liquid and cellular debris when accumulated in the alveoli undergoes damage, and such type of severe damage in the injured alveoli, widening of the edematous interstitium and the disrupted endothelial cell layer leads to more acute condition. Now the virus not only infect the lung, but if it damage lung, it can able to carry to other organs via blood. So other organ that can be affected by the kidney, intestine, blood vessel, heart, and brain also. And the psychological impact is very, very havoc, starting from anxiety to stress to panic to frustration and a number of things actually, and that creates the miserable life more miserable. Now long-lasting health effects was studied for the coronavirus 1 infection in Hong Kong. One of the studies showed that 2 years after the disease, 1 in 2 survivors, means 50% of the survivor, showed much poorer exercise capacity and healing status than those who had never had the disease. In another study, it was found that 78% of SARS patients can return to their full-time work after 1 year of infection. A third Hong Kong study revealed that 40% of the people recovering from SARS, they have chronic fatigue, may last for 3.5 years. And SARS and Epstein-Barr virus of herpes virus family, they both trigger chronic fatigue syndrome, which may last months to years. And long-lasting effect is the weakened heart, deterred lung capacity, brain and kidney damage, and deterred cognition. Now, the fatality rate of major virus outbreak in last 50 years, if we tabulate, we found that this virus, SARS coronavirus 2, do not have much more, the 2.2%, and now it is 2.2 to 3% actually, death rate throughout the globe, and a little bit lower in India. Now, compared to Marburg, which is 80% fatal, and then Nipah 77% fatal, even the SARS coronavirus 1 is 9.6% to 10% more, and the MERS coronavirus is 30.4%. Now I come to what we learned from the immunity side. I'm not going in detail to describe the immunity, because immune system is made, not built. So it contains several organs, and these are two immune system, innate and acquired. And just going to a strong immune system is the best antidote against this type of infection. As long as it runs smoothly, we do not feel that we have immune system. Just like a stagnant water, until and unless you throw something, you do not know that is water can create waves. So the interplay between the two wings, adaptive and innate immune system. You see, when a virus infects, the virus can be tackled. Whereas even in a bacteria also, the tackling mechanism is there. But if there is something wrong, then the acute inflammation takes place. And then if that inflammation goes to chronic condition or chronicity, then the damage done is much more, because the interplay between the two wings is a very delicate balance. If the delicate balance is unable to maintain, then only the havoc can happen, just like in some cases in this particular virus. Now, what are the imbalance? Why the imbalance in immune response against SARS coronavirus? Because the major host virus interaction, scientists have already learned that the delayed or suppressed type 1 interferon response during initial infection, in some cases, in the patient where the delayed or suppressed type 1 interferon response takes place in initial condition or initial infection, may be havoc. Viral replication trigger hyperinflammation condition. You see, influx of activated neutrophil, inflammatory monocyte or macrophage, and then Th1 and Th17 is induced and specific antibody are produced. Actually, what happened when the virus infect AC2, then type 1 interferon release was blocked. So due to such block, a lot of events takes place, and what happened, the innate immune system is supposed to control the things first. But innate immune system, sometimes what happened actually in this case, when the case is critical actually, innate immune system unable to control the virus, but it thought that it can do these things. So repeatedly they do the same thing, just like a good boy who is who is doing a mathematics, and he thought that he can able to solve the problem. But after several trial, he is exhausted. Here also, after several trial, the chronicity comes. And during that time, our adaptive immune system unable to get any signal from the innate system. So it releases cytokine like a storm, leading to the cytokine storm. I'm coming to describe these things. This is the predicted host immune response. It's a very complicated one. But upon entry of the alveolar epithelium, the coronavirus is recognized by several receptors called as innate immune receptors. They are the sensor actually. I'm not going to name in detail. This actually activates the NFKB, nuclear factor kappa B, and another interferon regulatory signal, and pro-inflammatory cytokine like IL-beta, IL-1 beta, IL-6, etc. Now, the antiviral activity of type 1 interferon is essential to limit the virus. But virus is so clever, it can block the interferon 1 release, and thereby cytokine releases by infected cell modulate adaptive immune response by recruiting and activating macrophage cannot take place. So what happened? The orchestra of macrophage, B cell and T cell cannot able to happen here, and to eliminate the virus. This imbalance immune response lead to hyperinflammation to severe COVID cases. Now, the latest report on interplay 2020, 5th May actually. Interplay between the immune system and SARS coronavirus 2 in COVID-19 patient. You see, when the SARS virus enters, it creates actually cytokine stress syndrome. First of inflammation, it creates inflammation and then creates secrete different cytokines like IL-1, IL-8, IL-2, TNF alpha, etc. Now IL-8 and IL-2 induces IL-6 and IL-17. You see, IL-6 can able to induce a protein in the amyloid protein actually in the liver that creates the fibrin deposition and to damage the liver actually. Now simultaneously, this cytokine storm or cytokine stress syndrome caused the activation of Treg cells that exhausted. Treg cells, if exhausted, then it increased the expression of TNF alpha due to over cytokine stress and that to thrombocytopenia. Now, thirdly, coronavirus causes endoplasmic reticulum ER stress also that activates the unfolded protein, which leads to prolonged ER stress with unfolded protein, activates NFKB pathway, which is very important for any viral replication, to inflammation and leading to the pathogenesis of airway diseases via inflammation. And so this is actually a lot of things collectively create the problem, and that interplay in immune system and SARS coronavirus need to stop to save the patient actually. Now, 20 to 50% of COVID-19 uninfected patients may harbor memory T cell, probably from the previous exposure to seasonal cold like coronavirus 229E, NL63, OC43, HKU, because these four viruses of corona, alpha and beta coronaviruses are circulating in our community and responsible for 15 to 20% of cough and cold. Memory T cell can cross-react with coronavirus too. So memory T cell already existed in some persons can cross-react with COVID-19 or coronavirus too, and can provide robust responses. So those people cannot go for developing acute symptom. They may be mild or may be asymptomatic. Pre-existing T cell immunity could only reduce COVID-19 severity, which we tried with, which we want to try with the virus vaccine also. With high level of pre-existing memory CD4 T cell, they recognize coronavirus 2 and can mount a faster and stronger immune response, exposure and disease severity and death, but unable to prevent infection. Now, memory T cell in achieving herd immunity, many will be infected, but fewer would be ill. It is called the herd immunity. Now, activation of cross-reactive memory T cell develop plasma and killer T cells will not provide immunity or protection or herd immunity, but mitigate or reduce the severity like many vaccines. Now, to control, how to control viral infection? Actually, in particular, the virus infection can be controlled. One is public health measures, second is vaccination, third is drug development. What is the public health measure in this particular disease? In this particular disease, we have to follow some COVID behavior. Behavior number one, we have to maintain a physical distance, whatever is possible. 3 to 6 feet is ideal. Now, we need to use the triple layer mask during talking. And now we are telling that triple layer double mask or a N95 mask. Avoid touching anything in outside and washing hands for 20 seconds every hour if it is possible. Now, these are the things that do not require any government. People himself or herself can do that. So health is a personal choice. As health is a personal choice, so please try to disseminate this information. Try to be to follow this behavior, COVID behavior to save your family actually. Second is the vaccination. Vaccine may tame several viral infections from smallpox to hepatitis. But you know, several viruses like influenza, HIV, AIDS, etc. cannot be controlled till death. Sorry. Here also, we have developed the vaccine. Now, the efficacy of the vaccine and the requirement of the vaccine that need further study actually this type of pandemic situation to prevent actually the infection. Our aim of the government is to prevent the infection as many possible in as many people as possible. Next is the drug development, prophylactic or infected individuals, therapeutic, prophylactic or therapeutic drug development. I will touch each and every point one by one. Now, to prevent transmission, first of all, scientists around the world, they try to understand what to prevent, what we want to do actually. How SARS coronavirus spread? They found that respiratory droplet, fomites, aerosol in the surrounding air. And in the closed room, in a closed room, the circulation of the air may contain the virus. So it affects lung and airways. There may be symptom or asymptomatic and may cause pneumonia to organ failure to death. So the cells responsible for Human Cell Atlas Consortium database showed that lung to nasal cavity to eye to intestine, heart, kidney, liver, everywhere we have ACE2 and transmembrane protein series 2. These are the major receptor of this virus. Now, viral entry receptor or targeted corresponding organ from nasal, lung, ileum, heart, eye, liver, everywhere ACE2 is there. So the virus can able to attack any of these cells. Now, treatment opportunity. What is the treatment opportunity? First is the development of vaccine to prevent the super spreading virus to do havoc. 10 are in emergency use with trial. Now some actually completed the trial and now in regular use. Repurposing of existing antiviral and some other drugs. Finding potent peptides and antibodies for neutralization. Phytophores from traditional recipe. Phytopharmacology, polypharmacology, and through the mode of studies on targeting host virus interaction, then computer modeling of drugs to accelerate drug discovery by artificial intelligence and machine learning. Now, vaccine development. You know, there are two ways. Classical method of traditional development of vaccine require 15 years or longer. And some of our vaccine already available requires 4 to 7 years minimum. But now, understanding from the HIV vaccine development protocol, scientists are well aware about the steps and they can overlap and jump certain steps. So SARS coronavirus vaccine development taken the short path, 10 months to 1.5 years total. So in that case, the trial regimen of phase 1, phase 2 and phase 3 are overlapping, 1 and 2, 2 and 3. And before completing the phase 3, the regulatory approval and other things are ready, and that's why within a very short period of time, here phase 1 and phase 2 trials was conducted simultaneously, and phase 3 after the interim analysis of 1 and 2 result, with several clinical trial stages running in parallel. Meantime, producer asked to start the large-scale production of vaccine candidate at risk. So, pathway to license of an emergency use authorization is not very clear, but still the things are going on. And the target is to deliver the vaccine as much as possible to the people very quickly. Now there are several processes of vaccine development. You know, HIV is one of the most challenging virus, and vaccine science have made enormous studies or enormous strides with this rapidly mutating virus. Years of experience on painstaking work researchers conducted to reduce timeline for developing COVID vaccine is now applied. And they have taken antibody route, chimpanzee adenovirus vector route, human adenovirus vector route, DNA conversion of viral RNA into DNA, and then messenger RNA of this particular virus. So these are the types through which the vaccines are produced, and there are several vaccines are in the pipeline, and some will be shortly, some will come very shortly also. Already we have 3, and in whole world it is 10. Now the coronavirus, we have AstraZeneca, we have Bharat Biotech, we have now Sputnik entered, and another is mRNA vaccine of Moderna, NIAID, and the Zydus Cadila, Zycov-D. So this is DNA vaccine and mRNA vaccine, and another is using the vector of adenovirus vector, taking only the viral spike protein. And the Bharat Biotech is the conventional vaccine taking the whole virus inactivated by beta propionate and then introduced into the body. So it can give the wide coverage because the whole virus is there. Now, the overview of the process of primary immune response. What happened? After entry of a virus or injected a vaccine first dose, the macrophage take up the virus and adopt the pathogen, undergoes phagocytosis. Pathogen is broken down into components of the phagolysome, and then these broken pathogen is exposed to the outer surface of the macrophage, which was recognized by the T lymphocyte. Now, T lymphocyte did two things. One, it direct the killer cell to directly produce antibody to kill these viruses, and as well as dictate the B lymphocyte. So B lymphocyte then proliferate to form plasma cells to produce antibody, and as well as it produces memory cell for future event. So it takes 7 to 13 days for the first response come. Now, after that, the antibody, specific type of antibody raise will be there, and the second dose actually gives you the specific antibody response. Now, due to this vaccine, second time when the virus or the exposure of the virus, actually what happened, if any viral disease now, if anybody infected with a virus, it is supposed that the whole life the person can get the protection, or that protection can be generated through the introduction of vaccine. Now, it happens to the classical viruses also, like pox virus. But in case of these type of short-lived viruses, because these viruses do not contain the full protein, they have glycoprotein or lipoprotein in their outer surface. Secondly, they are not going directly to the blood, they are going to an organ. So immune response takes some time to generate its proper response. So what happened in the second? So in the first vaccine, these are the either through the virus or through the vaccine. And in second cases, when you inject the second dose now, this whole event will bypass, and directly the memory cell produces specific antibody. Okay, and T cell also give the specific recognition of the virus. And same happen with other vaccines also. Now I will go to the next slide. We know what is antibody. Antibody is actually the protein produced in response to the antigen. So these are glycoprotein immunoglobulins produced in response to an immune reaction, and specifically bind to antigens that initiate the reaction. There are 5 types of antibody, and IgM, IgG, IgA, IgD, and IgE. They have 2 heavy chains, 2 heavy and 2 light chains. These light chains are, and the top part, upper part of these are antibody binding site and can change this configuration, and they are connected with the disulfide bond. Now, these antigen specific variable region have 2 antigen binding fragment that bind to antigen with high specificity, and simultaneously with 2 identical epitope antigen antibody binding of a particular antigen. Whereas the constant region includes the fragment crystallization portion to bind to the surface receptor and on circulating WBC, macrophage, natural killer cells, etc., to initiate an immune response. Now, these are the 5 antibodies, and they have this. IgM is a pentameric one. It has got 5 antigen connected together, so it gives the very main antibody for primary response and fixed complements. It acts as a B cell receptor also. In monomeric form, it involves in ABO grouping and RBC surface, but it enhances the engagement of the phagocytic cell also. It's a primary response. IgG is the main antibody for secondary response. It neutralizes toxin, opsonization, and provide long-term protection. Protect against bacteria, virus, neutralize toxin, bacteria of several bacteria, and trigger complement system also. It is actually these immunoglobulin may last longer time, 3 months to 6 months to 1 year also, and others are for different purposes. I'm not going in detail regarding this viral infection. Now I'm talking about the Indian isolates, specifically BELAGAVI isolates, or isolate from this Karnataka, of this part particularly. We have isolated some viruses here from the very acute cases. We take the viral RNA, convert it into cDNA, and then go for phylogenetic analysis. The tree analysis tree displaying sequence SARS coronavirus 2 strain to the assigned clade during analysis using a modern tool, Amwood Toolkit VIA, next strain server, and sequence strains are colored as per the assigned code with recent nomenclature. We found that two of our strains, NITM 1089 and National Institute of Traditional Medicine A1139, are marked with arrows. And we found that 8 changes are there, particularly 8 mutations are there, and these mutations are related to Maharashtra strain. So we showed how the strain entered the BELAGAVI region from Maharashtra. So these are the variants, and D614G, you know, is a common one and responsible for this havoc throughout the globe. That mutation is there, and this was published in a journal, Microbial Resource Announcement of American Society for Microbiology, and featured in the cover page article. Now, next strain classification of Indian strain so far showed that Indian strain has got a number of pangolin lineage, and that has got a number of mutations. I'll go to tell you some of the mutants, particularly single mutant, double mutant, and sometimes that third mutation is going on. Actually, mutation is inevitable in case of this type of single-stranded positive sense RNA viruses. But you know one thing, until and unless mutation goes to change the structure as it, neither the virus will be vaccine escape, nor there will be any damage. Actually, any more damage rather. While it is the tendency of anyone to change, and change is the name of the game. So virus undergoes change, and that change is functional change or functional mutation. So the functionality of the virus may change due to that mutation. Now recombination between the bat and SARS coronavirus pangolin and pangolin coronavirus resulted SARS coronavirus 2 due to cross-specific transmission. And mutation leads to several SARS coronavirus variants. There are many lineage. I'm just talking about what the people are telling about India. No, not this is India. It is available in 44 countries. And it was detected in October 2020, and later on global in 44 countries. There is B.1.617 lineage with double mutation and a specific mutation to another region also. It is still under investigation, and slight reduction in neutralization, and probably the virus is more readily can able to spread easily and invade the host quickly. So updated in March 21, these data, and if you are interested, you can go to search the data, because a lot of data are available and there are several changes going on. We are also working with this particular strain and trying to find out how many mutations are there in this particular area, from the taking sample from the acute cases, moderate cases, asymptomatic cases, and pre-symptomatic cases. Now, double mutant, what about the double mutant? You see, B.1.617, it has 2 mutations. In 484 amino acid, glutamate is replaced by glutamine, and 452 amino acid, leucine is replaced by arginine. So this actually undergoes stronger binding with the receptor and better shoot within the host, because this is the virus character. If a thief came into your house now, he or she will try to take maximum things they can. So it happens to be strong affinity to ACE2 is required for the virus to sustain, to cover up the whole spectrum of population. They have covered the older age. Now they are covering the middle age, and they will later cover on the child also in the lower age group. So, and another is proline to arginine mutation is there. These two collectively, actually what happened, one type of mutation, either this or that, is abundantly available. But what is important is that combining these two mutations in a single strain is the most important aspect. So proline to arginine mutation in 681 amino acid, they boost cellular inactivity by cleavage of S protein to active suit of S1 or S2. Now sublineage, this sublineage in India B1.6.617.1, 1.2 and 0.3. Now double mutation in B.1.671 occurs in spike protein with substitution in 484 and 452, two amino acids. But the term W mutant is misleading because in a short perpetual, viruses undergoes state of perpetual change due to continuous mutation, and it occurs globally. So emerging research suggests that variants may be more transmissible than the previous one, and this is the characteristic. If you go somewhere else now, you try to establish yourself there, and sometimes more aggressively. So, but the effectiveness of current vaccine against the variant is under investigation. But still, I am telling you, due to just change of some amino acid sequence, the functionality of a vaccine cannot be changed. Now, predictive interplay of COVID-2 in human health. A study showed that why the population from other countries are more affected, because bioinformatics study on COVID-614G subtype harbor an additional mutation of the S protein and clip site at S1-S2 junction, and that is specific to neutrophil elastase. Some population, Caucasus and non-Caucasus population, have genotype deficient of AAT deficiency, and therefore AAT deficiency is highly prevalent in Europe and North America, but very less in East Asia. Now, this AAT deficiency actually inhibitor of alpha tryptophan to prevent lung damage by neutrophil elastase that can prevent actually. So AAT deficient people have higher level of active neutrophil elastase, thus more susceptible to infection by 614G subtype. So this paper has been published by Bhattacharya et al. In Infection Genetics and Evolution in a few months back, actually, to 2020 March probably. Interested people can consult the paper, very interesting one. Now, the 6 months after the peak of first wave in September 2020, COVID-19 cases again rises, which we call as a second wave. Now, first week, signaling the arrival of second wave. More than 7 months, we got 8 to 10 months we got. But now the patients in both the waves actually, it was found that more than 70% patients in both waves are greater than 40 years of more than 40 years old. Only marginally higher proportion of younger patients. ICMR DG is that of opinion. He told that based on the study on 7,600 patients in the first wave and 1,885 patients in second wave, a higher proportion of asymptomatic patients are recorded in second wave with higher oxygen requirement. No major shift of age group and death rate in hospitalized patient. Although in some provinces the data may differ. So in COVID-19 symptom first wave versus second wave, if we see the higher proportion of asymptomatic and higher proportion of admitted patients showed breathlessness in the second wave. And patients with a sore throat and dry cough were higher in first wave. But now about 41.5% patients require oxygen in the first wave. And now 54.5% patients require oxygen in the second wave. Now prophylactic development. Now you see, developing a drug against a virus is not easy, I told you, because viruses are acellular nucleoprotein particle, and that nucleoprotein particle we have also in our genome. So if we design a drug against the nucleoprotein, that can cause the host also, that can target the host cell also. That's why most of the antivirals are toxic in nature. Now we have to find out two targets. One is the viral target, other is the cellular target. Here the virus target is spike protein, S1 and S2 domain, envelope protein, papain-like protease, PL-PRO, and nucleocapsid protein 3 chymotrypsin-like protease and RNA-dependent RNA polymerase. In case of cell target, cellular target is the lung cell pneumocyte 2, ACE2 and transmembrane serine protease, then goblet cell, and then ACE2, transmembrane serine protease, furin, cathepsin, and some other proteins, which are actually the other doors for this virus. Virus can not only go through ACE2, it can go through transmembrane serine protease 2, furin, cathepsin, and many of these. There are 7 doors actually for this virus to attach. What happened? It attaches with ACE2, and transmembrane serine protease and furin helps the virus to go inside in depth and establish infection quickly. So if we target any of these two, which are not very related or not very useful in human, so in that case, the virus can be tamed. So to control the virus, our target is to block the host virus interaction rather than targeting the virus alone. Now, current therapeutics. There are several therapeutics. Virus entry for chemostat mesylated, then virus fusion for chloroquine. Now it is not using now. And arbidol membrane-mediated fusion. Then replication blocker, leftovide. Then RNA-dependent RNA polymerase, remdesivir, ribavirin, and favipiravir. Now, WHO is telling that remdesivir, though developed for Ebola virus and it can work with the RNA-dependent RNA polymerase, but in case of coronavirus, it is not of very good use. So they are withdrawing the remdesivir from the list, drug list. Now, viral exocytosis by interferon alpha 2A, IL receptor and IL cascade by anakinra and tocilizumab, and decreased cytokine storm, we can use thalidomide, and for inhibiting cathepsin, that talcoplanin. Now, there are generalized home regimen for asymptomatic and mild symptomatic cases. You can use ivermectin or favipiravir, doxycycline, and vitamin C, D, and zinc combination, then acetylcysteine and levocetirizine plus montelukast or any cetirizine tablet along with, and if required, some in a close observation and under the clinical rather doctor's advice or clinical practice, people can use the other drug like methylprednisolone or steroid if required to inhibit the initial inflammation. Now, how ivermectin act? Ivermectin was studied in vitro study was done with ivermectin, and it was found that IMP alpha and beta 1 bind to coronavirus cargo protein in cytoplasm and translocate it via nuclear pore complex into nucleus, where it falls apart, and the virus and the viral antiviral response to enhance infection. Actually, this is done in the verocells were infected with the SARS coronavirus with an Australian strain for 2 hours prior to infection, but prior to addition of the DMSO as a control, or ivermectin as a drug control, ivermectin. Now samples were taken at 0 to 3 day post-infection for quantitative viral load and RT-PCR of cell-assorted virus as well as supernatant of the viral soup, and the infection was controlled or inhibited. It was shown. So far the ivermectin bind and disables the IMP alpha beta 1 heterodimer that prevent actually the binding of viral protein and enter the nucleus, resulting in efficient antiviral response. But in vivo study of this particular ivermectin is very difficult, and till date good data on in vivo study is not available. So this particular anti-parasitic drug is used in caution also. Another drug you are totally hearing about, 2-deoxy-D-glucose. You see, we all know that our metabolic pathway requires glucose, and in the first stage was glucose is converted into glucose 1,6-phosphate and then glucose 1,6,2,6 diphosphate. Now here hexokinase and then another isomerase enzyme is required. If the body produces 2-deoxy-D-glucose, but just eliminating one oxygen from hydroxyl group or the second position of glucose moiety, then what happened? This 2-deoxy-D-glucose cannot able to proceed further, and it reversibly block the hexokinase and isomerase enzyme. So the entire glucose degrading pathway or glycolysis will stop. Glycolysis stops mean energy will be less. Energy stops mean oxygen consumption will be less. Nearly 42% oxygen can be generated out of that. So oxygen dependency patient can be used with this one as a powder just, but 2-deoxy-D-glucose is not new one. For the last 20 years, people are trying or people are adopting this against cancer, and this was taken up by the DRDO, and they have developed this 2-deoxy-D-glucose, and they studied with SARS coronavirus infection, claiming that due to non-availability of glucose or due to glucose blocking of glucose degradation, the virus infected cell will not able to provide energy to the virus, so cell will damage and as well as the virus. But what happened to the live cells or other cells? Because when you take a glucose powder, 2-oxy-2-deoxy-D-glucose powder, it will go every cell except liver and kidney, because liver and kidney usually not take up 2-D-glucose. They have the selectivity. So entire body will take up. So there are a lot of questions and lot of issues are there. But in case of people, those who require high oxygen or high oxygen demand, in their case, 2-deoxy-D-glucose in controlled clinical setting can be useful. Now another is cannabinoid. You may heard that the people, those who are using cannabinoid or chain smoker are less affected by this COVID. Why? Because cannabinoid is a potent inhibitor of SARS coronavirus infection. There is different types of cannabinoid, and these are the spike protein positive cells and how they are tamed by this cannabinoid, supplier A, supplier B, supplier C. In every cases, cannabinoid can reduce the viral load and virus replication. You can go for this paper available in 2020-21 publication. And here I have shown how cannabinoid inhibits viral replication after SARS coronavirus entry into the host cells. In every cases, you'll see the declining of the virus titer and virus number by the CBD. They have used 10 micromolar concentration for 2 hours and then find that the CBD can able to control the virus also. So it may be one of the reasons the chain smokers or the people those use the cannabinoid, they can be controlled by, or they cannot be affected by this, or they are not affected by this viral infection, or less affected by this viral infection. Now,
Dr. Chattopadhyay,
yes,
can you a little sum up so that we can have some discussion? Because
yes. Now we are going from tradition to translation. Last part, we can block either ACE2 or transmembrane serine protease or furin, and the target blocking is several work is going on target blocking. Furin and transmembrane serine protease blocking is also there. We are trying with our process method of phytopharmaceuticals screening approach. We have developed an approach, a methodology using ELISA plate and then Western blot and several types of other methods also. Now, what we wanted to see actually, now there are several clinical studies going on, some phytoextract to create the society like a COVID-free, and omega-3 is another important food that can be used to tackle the virus infected people for better benefit. And now what society learned from it? Addressing COVID means addressing hypertension, obesity, diabetes, CVD, and chronic respiratory diseases. This is the opinion of Dr. Richard Horton, editor-in-chief of Lancet. Now, he is summarizing it. The COVID is not a pandemic, it's a syndemic. Pandemic highlighted the need of understanding the disease outbreak, whereas syndemic require understanding of implication of non-communicable diseases and its impact on the society or other societal groups. What lesson we learned from the society? 2 categories of disease interact in specific population. Infection with SARS coronavirus, and another is the array of non-communicable diseases. So syndemic is a set of linked health problem with 2 or more afflictions interacting synergistically and contributing to excess burden of the disease in a given population. So syndemic approach provides an integrated orientation of clinical medicine and public health to understand and treat the disease more successfully. Thus, holistic approach means mitigate the impact of virus and disease burden. But we have to consider the older population, their vulnerability, the Black, Asian, ethnic minorities, poorly paid workers. So no matter how effective the treatment or your vaccine, the biomedical solution of COVID will fail until and unless you target the society. So we need to devise policy and program to reveal or reverse the social disparity. Otherwise, society will never be truly COVID secure. So don't view the COVID-19 only as a pandemic, rather consider it at a multiple way to exclude the broader and necessary perspective, because society need hope, and economic crisis will not be solved by a drug or a vaccine, but national revival is required. Thus, accepting COVID is a syndemic will invite a large vision towards education, employment, and environment. Now, in future, what we have to, we may be more contactless interface interaction, strengthening digital infrastructure, better monitoring of intelligence and big data, then enable drug development through artificial intelligence, telemedicine, more online shopping, reliance on robot, because robots do not affected by the virus, and more digital events, even the esports. Now you may heard about the things that sex differ in COVID-19. Usually it was found that the ladies are less infected and the death has been occurred in women rather than men. So scientists are telling that biology to bad habits are responsible for that. Men involved in more risky behavior, ignore physical distancing, they are less serious, they have high BP, CVD, liver disease is more prevalent, and men have higher ICE2 in blood than the women. Whereas in case of women, they have stronger immune system because due to extra X chromosome, their immune system and immune response is better than men. You can consult these papers by Scully et al. In Nature Review Immunology 2020. And this is the sex difference in factors. They have shown how the innate and adaptive immunity of women are differ from the men, and viral entry and sensing as well as cellular response are differ. You can get it from another paper of Takehiro and Isawasaki in Science 2021. Now, take home message is we have to maintain physical distancing, 2 gaj ka duri, mask jaruri, and we have to use the mask whether you are vaccinated or not, it does not matter. Wash your hand with soap and soap water frequently, and jab tak we do not have any drug, preventive or curative, we have to follow the rules and regulation as WHO and all other authorities are telling. So with this, I think these are some of our publications in phytomedicine, in microbiology resource announcement, in Journal of Food and Drug Analysis, in Journal of Biomolecular Structure and Dynamics, and Frontiers in Medicine and Phytomedicines. These are the references. Thank you so much for your patience hearing. Nearly 1 hour. Over to host.
Yeah, thank you. Thank you very much, Dr. Chattopadhyay, for your excellent, elaborative deliberation here. And this is an August gathering, a platform of Karnataka Science and Technology Academy under Department of Science and Technology, Government of Karnataka, is a platform for popularizing science as such to among the masses who are having the science in their passion and in their mind. I think such type of talk at this moment was very effective and important. So I must give thanks to Dr. A.M. Ramesh, who is Chief Executive Officer, for selecting such topics for the discussion at the moment where scientists, doctors are enhancing and physical on all the healthcare people, professionals are working together, including administration, government, for how to overcome this pandemic situation. The talk was excellent in the way you have started with the deliberation by structure of virus to its etiology, to its manifestations, site of actions, changing the pathophysiological structures and pathophysical orientation, immunological aspects you have entered and how it affects, and from there to how it can be actually the viral domain and the types of different virus, including the mutation format, to the development, how to control and treatment in this way. We have explained to the drug development, to vaccine, specifically the aspects of the vaccine, to other ways of the biochemical aspects we have discussed in details, and lastly the hope and what we're supposed to do, also given a societal aspects of the entire talk. As a nutshell, I should say it's a brilliant, because that's required for all the peoples who are listening today, is a platform of the Karnataka Science and Technology Academy. It's a really important and interesting. I think that as a technically, I supposed to ask one questions because I was chairing the session. So I will ask and then I will see that other people can get a scope to ask the questions. I will over to Dr. Ramesh sir. So my question is that in my own understanding and my own actively involving in the clinical medicine of the COVID aspects, because we have 500 bedded COVID hospitals in our university teaching hospitals, and we know very well how it is going on in the first waves and second waves, and specifically the second waves after its arrival in the March 3rd week onwards, the situation deterioration and again little better as compared to the last week. Now every day and every week counts. The different aspects we are finding for management of COVID patients, specifically in the critical care management where the respiratory supports or oxygens and respiratory supports are very much required for the patients. So we are concerned of one important aspect, that is the use of steroids and use of anticoagulant drugs. The timing, if it is missed, neither steroid works nor anticoagulant work, or timing it is wrong, both of them are to be disaster. And these aspects I think need to be done. What exactly you think of it? Because steroid compromises immunity and already virus immunoescaper. So how these things can be better suited and how people should not misuse such steroids arbitrarily, because this drug is available in the market for different reasons, purpose, not only for the COVID-19, but people think that purchasing the blood thinner like ecospirine or aspirin or the steroids, which is a normal drug, and some antibiotics, azithromycins, doxycycline, they already procured and kept it, but they are not doctors. They are not the professionals to use it. This is one problem we are facing when we are treating the patient. Patient come in the hospital, going to the critical care, including up to 60 liters per minute, we are flushing the oxygen in a patient in such a critical care with a high COVID score up to 23, 24 level out of 25 we are getting in the CT scan, high resolution CT scan it goes. And many of them are said that they have already taken the steroids in their home. So this is one of the issue. Kindly tell a few words about that justification and judicial use of steroid for COVID as such, and how it should be controlled for the common interest of the people who are not doctors, maybe scientists, but not doctors. Sir. Yeah, please unmute. I think you are muted.
Thank you, Professor Das. It's a very, very important and very pertinent question you raise. You see, I feel that steroid or use of any drugs should be under clinical or should be under a doctor, under clinical supervision. Neither in home nor in any other setting except the hospital setting, steroid can be used. Because the use of steroid, if the time is wrong, yes, the patient cannot survive. And if survive, the patient may suffer into mucormycosis and all other diseases. Okay.
Right.
So this is number one. Number two, steroid use and use of this type of drug combination is for saving the life, not just mitigating the disease. Maximum what disease can do can make a person sick for few days, maybe 14 days, maybe 21 days, depending upon the amount of virus or viral load in the body. Reaction. If a person, I feel that if a person have fever consistently for 4 to 5 days, maybe 99, maybe 100, he must go to a doctor first. Test it and follow the doctor's advice. But not be with after 3 to 4 days is maximum time for any change or any symptom in your body in this pandemic situation. It is better to talk to a doctor and do that work accordingly, not taking any decision or purchasing any medicine and take it. Because if you take an antibiotic without any precaution, your bacterial flora may be affected on it and maybe there are some problems. If you take any other any other drugs except the say fever reduction aspirin type of drug. All the drugs have side effect as well as effect. Those who has effect, it has also got side effect. So self-medication is totally prevented and it should be stopped. This is number one. And the advisory should tell like this from the government sector also. And regarding the use of these other drugs, particularly the prednisolone type of drugs or the steroid type of drug, is only on the medical supervision, otherwise not. And secondly, the situation is just like this. Mass media is propagating like anything, and they are telling the name of the drug also. So people are going to a doctor, or say for example, one of my relative was infected, so I got the prescription from him and I procured the same drug. So this is going on. It should be stopped. Right. I think the science organization can take a lead on these things, what to do and not to do.
Absolutely. There must be a message from the scientific forum among the common public, I think, that the rational use and very justified use of the drugs, this is not the situation as a normal, because the wrong use and bad use and misuse may make the disaster. We are suffering it in the hospitals because patients are in a critical and we are unable to control them. The reason is they are already taken the drug when actually virus are proliferating. When virus in the proliferation stage of the initial, they have taken the steroids, and now we can't do anything at the moment when it has gone too serious, too severe and extremely critical. I think it's right. So I request one second to Professor Dr. C.M. Ramesh sir. Sir, you take over the mic from me now, and I finish this responsive. Thank you, sir.
Very grateful to you, sir. I have a few questions myself. In fact, I have attended 4 or 5 talks on COVID-19. By far, this is the best that I have attended. Congratulations, sir.
Thank you, sir. Thank you
for your excellent talk. And I have few doubts myself. If you could permit me, I shall ask.
Yes, sir. You can.
First one is, yes, sir. Now, there are 2 types of vaccines, DNA-based and RNA-based vaccines. Which is more effective for COVID-19? That is a very good question.
I'm just completing.
Okay. Will I answer it?
Yeah, please, sir. Please, sir.
Okay. Actually, efficacy of a vaccine do not depend on how it produces. It depends on what it delivers. Okay. Yeah, so correct. These are the methods: developing the vaccine via DNA route, or via RNA route, or via a vector route, or the whole virus route. Actually, it used to say the effectivity and efficacy. When a vaccine undergoes trial, they are doing the trial with 2 sorts of 2 arms actually, placebo arm and the vaccine arm. Now with a minimum population. Now when the vaccine is delivered to a mass, then that efficacy is different. So effectiveness and efficacy are different. Therefore, we have to see which vaccine can able to protect maximum number of population. Okay.
Yes, sir.
That we have to see. Okay. So not the type of vaccine. Vaccine are of different types. There is no problem. But one saying goes on. If you know the Edward Jenner developed the vaccine first, discovered the vaccine procedure first. He take the whole soup of cowpox and then given to a boy. And that is the remarkable history creates from that day. Here also, if the whole virus vaccine is there, probably that can able to give wide coverage. May not be effectivity much more, but coverage will be much more.
Yes, sir.
So 2 things you have to remember. How many population are getting protected from this? Some data is available. Sputnik has covered much more than other vaccines, and Covaxin is giving more wide coverage to tackle all types of mutated viruses.
Yeah. Sir, my second question is: plasma therapy helpful for COVID prevention?
Actually, no. Plasma therapy is not for prevention. Remember, with plasma, you are getting a number of things, blood proteins also. And that is not the way. only for saving the dying patient. It can be used. Now ICMR is withdrawing that also as per WHO also. So plasma therapy is not the answer, and don't go for plasma therapy as a preventive measure.
Yes, sir. I have two more questions. We talk
Yes,
about
sir.
herd immunity. We talk about herd immunity. After vaccinating, how much portion of the population herd immunity can be developed?
Actually, until and unless we cover 70 to 80% of the population vaccinated, it is very
Yes, sir.
difficult to get herd immunity. Okay.
Yeah. Okay.
Either by infection
Sir,
or by vaccination, we have to cover
yes, sir.
nearly 70 to 80% population.
Oh, yes, sir. Sir, since the beginning of COVID infection, there has been talk that it is not natural, but it is a man-made virus. Is it true or not?
Actually,
There was a big
yes,
there was a big article in the week also that it is not natural, but it is a man-made. I want your words on that.
your question is very tricky question. You see, a lot of debate is going on. As a virologist, you can say I am a viral biologist. As a viral biologist, I can say that this type of virus can produce by recombination, and that recombination can take place in the animals like bat. And if it is man-made, then man always made mistakes. There are several examples. Until recently, the virus do not show any hints of any man mistake. So it's very difficult or very unlikely that this virus is created by human being, maybe researched by human being, maybe leakage is there, but man-made virus is not. It is not a man-made virus. That much I can tell you.
Yeah, because there was a big article on in the
that is
week about this. That's why, sir. Once again, I'm very grateful to you, thankful to you for an excellent, illuminating, and informative talk on COVID-19, a lesson for science and society. In fact, this is the best session that I have attended. Thank you very much for you.
Thank you so much, sir.
I'm very grateful to Dr. Kushal Das sir for suggesting you for this talk and also for chairing this session so well. We are very grateful to you, sir.
Thank
And
you.
sir will talk on indigenous antibiotic plants in North Karnataka in the perspective of COVID-19. We look forward to your talk, sir. Until then, thank you very much. I'm also grateful to our honorable KSTA member, Dr. Ambekar Eknath, sir, for attending this session. And many senior faculty and academicians have joined this session, including Dr. Satnoor, Dr. Ramakrishna Reddy, Dr. Manohar Dadmi, Dr. Tabasam, Dr. Srinivas, Dr. Venkappa, Dr. Balbir, and Dr. E. Sagar, and also many of my young friends have joined this wonderful session. Thank you all for you for joining this session. We look forward to your kind encouragement for our programs. Next week, Kushal Das sir will be delivering the distinguished talk. I request all of you to join this session and also thank my colleagues, Mr. Umesh and Dr. Anand, for being with me in organizing this program. Thank you very much, sir. Grateful to you once again. Thank you. Thank you, sir.